AI. And we are presenting today on this collaboration that we did. For those who don't know, we worked on a biomodel and that does protein engineering and has some interesting results that we'll go through. This is work that happened mostly end of last year into this year. We announced this in August. And so we'll be sort of going through the results both on the ML side and the bio side. To start, Rico, I'll let you share some of the context.
Hello. Yeah, so as John mentioned, if I tell you we're doing protein engineering for longevity, you might want to be like, why would you do protein engineering? Everyone in the field uses small molecules or combinations or knockdowns of genes. So I wanted to take a step back and go back to the GLP-1s. And to me, this is essentially a story of 23 years of protein engineering, because we started with the knowledge that the human GLP-1s, which are secreted after you eat food and give you this feeling of satiety, only have a half-life in the plasma of about 1 to 2 minutes. So they were secreted and basically immediately chopped up by this DPP-4 enzyme. So about 23 years ago, we discovered this mechanism, or 23 years before we actually got to the drugs, we discovered this mechanism that this GLP-1 analog from the Gila monster has a half-life of 2 to 3 hours because it doesn't have this DPP-4 cleavage site. So even though we knew this mechanism to essentially make a more stable GLP-1, it still took us 23 years to essentially engineer liraglutide and semaglutide, which have the sort of half-lives that we know and love today in the drugs that people are taking. And this is essentially a story of 3 amino acid changes and attaching a fatty acid side chain to have higher stability in the plasma. Okay, so I hope I was able to lay out the context for protein engineering being a very powerful tool to engineer very potent drugs for very large disease areas. So how does this link to longevity? And to take a step back, as most of you are probably aware, there's these 4 proteins called the Yamanaka factors, O, S, K, and M. And they're able to essentially take any cell type in your body and turn it into an induced pluripotent stem cell, which is sort of like the equivalent of an embryonic stem cell. And it was found that when you redifferentiate those cells into, back into a skin cell, for example, that you now have a rejuvenated skin cell, or at least a skin cell that has all the sort of hallmarks of what a young skin cell looks like. And it was also found that this process can modestly extend lifespan when you overexpress them in the mice. 12.5% is interesting, but not great, because we have much more, much stronger lifespan extending interventions that are already known. So why is it only 12.5%, given that the mechanism of rejuvenating cells and tissues should give us a lot more than that? And one of the sort of what I like to call is the dirty secret of reprogramming is that it's very slow and inefficient. Only 0.01% of the cells that you're reprogramming in a dish would actually turn into these pluripotent stem cells. And additionally, there's a strong negative correlation of reprogramming efficiency with age. So here on the x-axis, you see the donor age. On the y-axis, you essentially have a proxy for reprogramming efficiency. And you can see that this goes down to the point where at an 80-year-old donor cells, we're not able to reprogram them at all anymore. So how do we fix this problem? So there's a leading academic effort which came up with this super SOX protein. And that was essentially 15 years of rational protein design and swapping domains to merge these 2 proteins, SOX2 and SOX17, to create super SOX. And super SOX was able to get a 50x increase in reprogramming efficiency, which is really promising. But all of us, we want to solve longevity within our lifetime. So we don't have 15 years to engineer a new protein to then test it in clinical trials for 10 years. So we were really asking this question, how can you do this a lot faster?
And so this sort of motivates why we started working on this model that we, in my opinion, poorly named GPT-4B micro, but sort of the core idea is we wanted to try to train a large language model, but specializing specifically to be useful in protein engineering tasks generally. So this is not specifically targeting OSKM or longevity proteins, just sort of having the ability to generate, edit novel proteins, and make them useful for various tasks. The way we approached it was to sort of like start by thinking about what makes LLMs in general powerful. And I think the strong case we made is that sort of the generality of pre-training on a large diversity of data sets gets the models the ability to sort of cover a lot of different types of data and draw relations between them. And so we trained on literature, structure, which is tokenized using encoder decoders, coevolutionary sequences, protein interactions and bindings, families of proteins, and various sort of like data augmentations combining these. And the result is a model that sort of, we'll talk a little bit about like how well it works, but sort of just covers a little bit of everything in terms of what you could expect an AI model to be able to do with regards to proteins. So it folds, it does inverse folding, it does generation, we can communicate it with natural language, and we train it with these like very nice clean scaling laws. We like bladed different data sets at small scale before doing a bigger run. And so the result of that is just a model that we found to have like a very broad set of capabilities. We trained it with long context using multiple sequence alignments to be able to increase like the steerability of the prompts that we used when generating models, which Rico will talk a little bit later. And the result of that is that we had this model that was very small, very specialized for biology and proteins, but impressively strong on a lot of various biology evals, even compared to sort of like at the time frontier reasoning models. It is decent at all the various tasks. So if you compare it against like ESM fold models that are specialized for protein folding, 4B micro does decently. And this is sort of with it being a relatively small part of the data set and a small part of the capabilities of the model. We looked at the attention maps. It behaves similarly to prior models, sort of with like attention mappings between residues, mapping roughly what you would expect. But sort of the core takeaway from this is we developed a model which we tried to make as general as possible, trained on as much of biology and proteins that we were able to gather with open data. And we see sort of similar to in large language models based on text and other modalities that they work quite well, they're quite general. In silico evals were quite interesting. And so this excited us and inspired sort of trying out the model's generation capabilities in vitro labs, which Rico will talk more about.
So our process for testing this model's ability in the lab was we essentially had it print a bunch of protein sequences, and we ordered like 50 to 100 of those, produced them as DNA, dropped a bunch of fibroblasts, which are these skin cells that we use for reprogramming. We programmed them towards an iPSC state, and then sort of looked at which variants performed best. So I'm only going to briefly touch on those results. I would refer you to the blog post for more detail. But we essentially started by doing a pilot screen, which was we took our best human scientists within Retro. These are people who have experience with reprogramming over like years, of course. And we sort of asked them to design a better RetroSox, RetroKLF variant and see how well they would do. And they did somewhat well, but probably around the same efficiency you would get for the wild-type OSKM. When we asked our model to redesign those sequences, we saw a massive jump in these early and late pluripotency markers that we would want to see for more efficient reprogramming efficiency. So that was really promising. So we went and replicated those results, this time measuring whether the proteins are actually able to take those cells all the way to iPSCs and them exposing like actual iPSC markers. And you can see at day 10 in our control, which is OSKM, we didn't see any of those colonies. What you want to see here is a bunch of like blue like colonies of cells. Whereas for our factors, we already saw like quite a few of those emerging. So the reprogramming process is not only more efficient, it's also a lot faster than the standard OSKM variants. The next thing that we want to test, because for any sort of like in vivo application, we really care about not just the reprogramming, but also the rejuvenation aspect of OSKM. So one proxy of that we used is essentially their ability to resist DNA damage when you treat them with a chemotherapeutic drug. And you can see that OSKM, the control, our positive control here, does somewhat better than GFP, but not very convincingly better. Whereas our factors are able to significantly improve the cell's ability to withstand DNA damage upon treatment with a chemotherapeutic drug. So yeah, what we're really excited about is that we were able to take something that was previously a 15-year sort of protein engineering endeavor and break this down into like a 3-month process of having these very potent reprogramming factors. So I just want to zoom out in the end and like leave you with another message. So I mean, we're really wondering, like, how can we actually like achieve lifespan extension with the tools that we're using these days? And to me, this project really shows that protein engineering is potentially a great sort of like substrate for engineering new drugs for lifespan extension. And I started my story with talking about the GLP-1 receptor agonists, which already we have some data is starting to trickle in that these may be the first FDA-approved anti-aging therapies. This is a plot from a study where they looked at obese, non-diabetic people, and we see a reduction in all-cause mortality. And I'm sure over the next couple of years, we'll see more data coming in. This is really exciting for the field. The other thing is that the most potent lifespan extension known thus far is this result from 2008, where essentially it took the H1 gene and introduced a single mutation or a protein truncation, which in essence is a way of protein engineering. And the truncation was able to achieve the largest yet known relative increase in lifespan extension, which is about 9x. So to me, it's like the first human anti-aging drug and the largest known lifespan extension effect to date have both been achieved via protein engineering. So I guess a provocative question I would pose is that, what if the key to really extending human lifespan is just out of reach for the traditional tools that most of us in the field are using these days? Thank you.
Awesome. Thank you so much. Questions, comments?
So I saw that and I've heard that this GPT-4B micro is capable of being interfaced with via natural language. I was wondering if there's any issues with consistency in terms of outputs and whether subtle changes in the phrasing of how one might interrogate this model for protein design may influence the types of protein designs that are outputted as a result.
Generally, small changes in prompting can lead to big changes in output, but when you say specifically inconsistencies, what did you have in mind?
So I'm just thinking, like, from a very systematic perspective, if you want to get the best protein for X function and you say, create this protein to do X, or I would like a protein to do X, you may get a completely different design just based on the fact that you said create versus I would like to. And it seems like there could be a pretty wide space of possibilities that you would have to explore in terms of optimizing prompting things that really shouldn't actually matter that much. And so in order to actually get the optimal result.
Yeah, unfortunately, LLMs are not magic, and yes, prompting does matter a lot. You can get very different results depending on prompting. I don't know if you want to talk more about it, Rico.
I mean, one thing is, I think when John and I talked about these results after, I think we're sort of more, like, this is maybe like the GPT-3 moment of biology, where it's like, your model can sometimes get interesting results, but you still have to put a lot of effort in like engineering your prompts, and like most of it will be garbage, so you need good filters afterwards. But we do think that there's a lot of alpha to be gained by even just like post-training this model, because this is a pure pre-trained model. Yeah, so. I mean, to your point, there's definitely a lot of engineering to be done to get good outputs right now.
Sure. Is there, sorry, one more follow-up. Is there any like consistent, do you guys have like a set of recommendations or something for trying to sort of systematize the prompting as of yet, or are you moving towards that?
My sense is that this is a very novel kind of model, and prompting has not been figured out at all. Yeah.
To that end, would you care to share any prompts or fragments of prompts that are given in natural language to these protein engineering models, the MycoB, or and also that ninefold increase, what was the organism? I realized that was a separate
C. elegans.
C. elegans, okay, thanks. Anything about the prompts? That, like, I'm just curious, what the heck did they look like for this kind of a model?
Oh, I mean, the way that we were doing it is we were essentially showing it like proteins across evolution, like how they evolved, and sort of like asking the model to forward simulate the evolutionary process. Like you can imagine giving it like the SOX2 of a pig or like a chimpanzee, and then asking it to generate what it thinks should a human SOX look like, and then sampling a bunch of those and selecting for certain properties, and then just testing in the lab. So I think like showing the model a bunch of sequences that could contain an interesting pattern. You could also run a screen, take the best ones you got and show it to the model and have it figure out the patterns among the ones that perform best.